In this lecture, we are going to talk about patterns of inheritance for the first part, and then discuss the details of the gene expression. So before 20th century, there were two basic concepts in terms of the ideas of how the information is passed from the parents to the offspring. First concept was that heredity occurs within the species, and the second concept, which was also correct, was that traits are transmitted directly from the parents to the offspring. So the main problem was that nobody knew how it worked. Nobody knew what was the basic unit of hereditary information. And the second problem that arose from that understanding is that if traits are constantly blended within the population, why we can still see people being distinct. So eventually the term gene was introduced. Gene is the unit of hereditary information that encodes some of that hereditary information to a form of a particular trait. However, we must say that eventually it was revealed that some traits, phenotypical traits, require more than one gene for their manifestation. Now, every gene is located in the locus. This term means the place of a gene on the chromosome. And genes within chromosomes exist in the form of alleles. If you look at the maternal and paternal chromosomes, then each of these two will carry the gene. Maternal will carry one allele for the gene, and paternal chromosome will carry another allele for that gene. It is important to note that both these genes encode the same protein, but they can be slightly different, and one allele can dominate over another. So if alleles on both chromosomes, on both homologous chromosomes, are recessive or both are dominant, so both are recessive or both are dominant, we are talking about homozygous presentation, homozygous recessive or homozygous dominant. If one is dominant and another one is recessive, we're looking at the heterozygous genotype. So here, first of all, you see the example of a locus for different genes. So the M locus here in this example contains the gene that determines the leaf color. And you can see that both chromosomes carry the same allele for the M gene, they're both pink. Now, D gene determines the plant height, and you can see that both chromosomes, both homologous chromosomes, carry the same allele for the D gene. So this plant is homozygous for the D gene. But if you look at the BK locus, which determines the shape of the fruit, one allele is different from another. So that would be an example of heterozygous genotype for that particular gene. So the person who basically discovered homozygous versus heterozygous genotypes and phenotypes was the monk Gregor Mendel. He was experimenting with pea plants, and he produced true breeding strains for each trait. True breeding means that all plants were either homozygous dominant or homozygous recessive. And interestingly enough, not only this, what it means is that, for instance, if in certain plant the peas were smooth, they were smooth at every generation in every plant. If they were shriveled, they were shriveled in every generation for every plant. So then he started to cross-fertilize true breeding strains. So this term, alternate forms of a trait. So let's say white flowers plus red flowers, just as an example. Now, reciprocal cross in this case. So reciprocal cross is the, for instance, he was crossing red male plus white female plant, or he tried the other way. He did red female plus white male plant, just to make sure that sex does not contribute to the outcomes. And once he produced the offspring, which is a hybrid offspring after the crossing of two true breeding strains, he allowed that these offsprings of true breeding strains to self-fertilize several generations. And for each generation, he counted the number of offspring showing each form of the trait. So in other words, he started with, just to illustrate it, true breeding plants. Crossing them, he produced F1, and then by allowing F1 to self-fertilize, he essentially generated F2, F3, etc. First of all, let's define what a monohybrid cross. So in monohybrid cross, we are talking about only one trait and two variations of it. So for instance, we mentioned before red versus white plants. Observing only this trait and only two variants of that trait is an example of monohybrid cross. So Mendel, in his experiments, managed to cultivate pea strains with 7 different traits, and each trait had only 2 variants. So he could produce monohybrid cross for 7 different traits. He was looking at the flower color. He was looking at the color of the seeds, the texture of the seeds, the color of the pods, the shape of the pods, the position of the flowers, axial versus terminal, and the height of the plant, tall versus short. When he started looking at the monohybrid crosses in F2 generation, the generation, remember this one, so he started with true breeding plants, then produced F1, and then by crossing inside of F1 generation produced F2. So he observed that the ratio between two different alternate types of that particular trait were always fairly close to 3:1. What does it mean? Well, let's go back to the story of how Mendel produced his plants. So he crossed two true breeding strains, and all plants in the first filial generation, all F1 plants, resembled only one parent. So this resemblance of one parent is the dominant. So for instance, purple flowers. Purple was dominant to white. There were no white flowers in the first generation, only purple flowers. And another important thing, that there were no plants with the characteristics that would be in between the characteristics of the parent. For example, there were no plants that would be neither purple nor white, but let's say, I don't know, pink. As you may remember, the F2 generation was produced after the self-fertilization of the F1 generation. And in F2, the trait that was hidden in the F1 generation suddenly reappeared, not among all F2 individuals, but among some of them. And Mendel discovered that the ratio in which dominant and recessive traits appeared in the F2 generation was 3:1. So this is an example of what was happening in the first and second generation of the plants. Let's start with this. True breeding purple flower plant and true breeding white flower plant, they hybrid in the first generation produced only purple flowered plants. However, when the first generation offspring, F1, the purple flowered, self-fertilized, the F2 plants had 3 purple flowers and 1 white flower. Now, phenotypically, the ratio is 3:1, but the genotypic ratio is actually 1:2:1. So this was the dominant to recessive ratio as it appeared in terms of the phenotype. But later Mendel discovered that out of these hypothetical 4 plants, 1 was actual true breeding dominant, 1 was true breeding recessive, and 2 were dominant, but they were not true breeding. So this was the ratio between those plants. So what were Mendel's conclusions regarding his results? First of all, he realized that plants didn't show intermediate traits. Second, he realized that for each pair of alternate forms of the trait. One trait was dominant and the other recessive. For example, purple was dominant over white color of the flowers. And the pairs of these alternative traits that he examined were separated, segregated among progeny of a particular cross. That may sound very generic, but the idea was this, that it's not a blending of traits, that each individual progeny receives a specific combination, either becoming true breeding dominant or homozygous dominant, true breeding recessive or homozygous recessive, and non-true breeding dominant, which essentially is now known as heterozygous. And here, alternative traits expressed in F2 generation 3:1, dominant to recessive. We have to remember that we are talking in this case about the phenotype. Now, let's take a look at the model of inheritance that was proposed based on the Mendel's work and later discovery of genes. So it was proposed that parents transmit heredity in the form of discrete factors named genes. And each individual receives one copy of that gene from each parent. We call it an allele. And not all copies of that gene are identical. Alleles are not identical. And as we mentioned before, the alleles in the same individual come from two different parents. If alleles are identical, the individual is called homozygous. If alleles are not identical, an individual is called heterozygous. Now, alleles within the individual remain discrete, they don't blend. And the presence of an allele doesn't guarantee its expression. If there is a pair of two alleles, one of them is dominant and one of them is recessive, only the dominant allele is going to be expressed. And that explains the difference between the genotype and the phenotype. As we mentioned before, the ratio of genotypes in F2 of Mendel's experiments was 1, 2, and 1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. On the other hand, the ratio of phenotypes was 3:1, because both homozygous dominant and heterozygous individuals demonstrated dominant phenotype. So the principle of segregation guides the inheritance in the sense that the alleles for a gene, two alleles for a gene that come from a single individual, they segregate when gamete is formed during meiosis, and then randomly rejoin when one allele comes from each parent during the fertilization and the zygote formation. And the physical basis for the segregation of the alleles is the independent assortment of the chromosomes during meiosis. It's remarkable that Mendel had no idea about this whole concept of independent assortment of chromosomes during the metaphase 1 of the meiosis, but yet he managed to identify the basic guiding principles of the inheritance. So in metaphase 1, independent assortment is responsible for the distribution of the alleles. So as we discussed in the classroom, the Punnett square allows us to identify which possible genotypes can be found in the offspring of two organisms with a known genotype. So in this example, the plants with purple flowers and the plants with white flowers were crossed. Purple flowers is the dominant allele. White flowers is the recessive allele. The dominant allele is usually written as the capital letter, and the recessive allele is written as the lowercase letter. So true breeding plant that is white flowered is homozygous recessive. True breeding plant that is purple flowered is homozygous dominant. And this is an example of the heterozygous purple flowered plant. So here you can see a heterozygous parent which produces two gametes that contain both the dominant allele. This is the heterozygous parent that produces two gametes. One of them contains the dominant allele and another one contains the recessive allele. So in the Punnett square, when you start filling it up, this is what it looks like. So you have two parents, each of them is heterozygous. So each parent can produce one gamete with the dominant allele or a gamete with a recessive allele. So possible outcomes of that cross will be one homozygous recessive, two more heterozygous, and finally one homozygous dominant. So when Mendel was crossing two true breeding plants, shown here, the purple parent, which was true breeding purple parent, which means homozygous dominant, generated two gametes, each of them containing a dominant allele. Homozygous recessive parent generated two gametes, each of them containing the recessive allele. That resulted in the first generation containing four heterozygous plants. When those heterozygous plants self-fertilized, it resulted in one homozygous recessive plant which produced the recessive phenotype, and three plants with the dominant phenotype. But out of these three plants, one was homozygous dominant and two others were heterozygous. In case of human traits, it's a little bit more complicated. Some human traits are indeed controlled by a single gene and may show dominant recessive inheritance, and you can track the inheritance patterns by using the pedigree analysis in families. For example, a dominant pedigree is juvenile glaucoma. This condition causes the degeneration of the optic nerve, and this is the dominant trait that appears in every generation, well, because it is dominant. Here are some examples of the human traits, dominant and recessive alike. For instance, albinism, lack of pigmentation of melanin, alkaptonuria, inability to metabolize a particular acid, color blindness, cystic fibrosis, muscular dystrophy, hemophilia, when blood cannot properly form clots, and sickle cell anemia are all recessive traits. So they may or may not appear in a particular generation, but these traits, such as mid-digital hair, short fingers, Huntington's disease, it's a neurodegenerative disease, PTC sensitivity, inability to straighten a little finger, camptodactyly, hypercholesterolemia, it's human Mendelian disorder, and polydactyly are all dominant traits. So now here is an example of what's called a pedigree analysis. So you can see that there's an affected male and unaffected female, and the offspring is either unaffected or affected. So if the male is, and then in this case, in the case of other mating here, you can see that the mating of the affected male with unaffected female and a mating of affected female with unaffected male will produce affected offspring. So it means that this one in the first generation is heterozygous, unaffected is homozygous recessive, so the possible outcomes are homozygous recessive and again one more heterozygous. And you have to appreciate that it's a probability, so when two individuals like this mate, it's totally possible to have a heterozygous dominant in inheritance. And in this case, also heterozygous dominant. So for the recessive pedigree, example is albinism, when melanin is not produced. In this case, the albinism is because of the non-functional allele of the enzyme called tyrosinase, and it equally affects males and females. And affected individuals have unaffected parents, which explains why it's recessive. In unaffected parents, they are heterozygous, but the affected offspring here is homozygous recessive. So here you can see that one of these parents here is heterozygous. So they do not show the pedigree, they do not show the trait because they are heterozygous. But when the offspring remains heterozygous, but now they carry the trait, and then when two carriers essentially mate, they produce homozygous recessive offspring, homozygous dominant, and the carrier heterozygous offspring. And same here, you have heterozygous that mates with homozygous dominant, so they produce two homozygous dominant offsprings and one which is heterozygous. And eventually, two carriers, two heterozygous parents produce homozygous recessive offspring. So now let's give a definition of the dihybrid cross. In dihybrid cross, the idea is that you look at two separate traits, and it's important that the traits are separate. It means that the inheritance does not depend on each other. So dihybrid cross produces, in dihybrid cross, when Mendel was studying dihybrid cross, it generates true breeding lines for two independent traits. So here, for instance, one line is true breeding for both shape and color, and another line is true breeding for recessive forms of both shape and color. So the first generation, when these two organisms, these two true breeding plants are bred, first generation only shows the dominant phenotypes. But then, just like with monohybrid cross, Mendel allowed F1 to self-fertilize to produce F2. So F1 self-fertilizes, and now we have two organisms which are heterozygous for each of the trait, and the ratio of four possible phenotypes is 9:3 to 3:1. Now we're going to take a look at the distribution of this. Okay, so here, what you're going to observe is this. So it all starts with true breeding plants, true breeding for each of these two traits, and they produce essentially heterozygous plant, heterozygous for each of the traits. So due to the independent assortment, these are the possible distributions of the genes, dominant or recessive, within the gametes. In other words, there can be four different gametes. And let's take a look what happens in F2 after the cross-fertilization of the F1 plants. So these are the possible combinations, and you can fairly clearly see that there are several distinct phenotypes. So one phenotype are smooth, round, yellow peas. In this case, it's dominant for both of these phenotypes. Then, and more here. Then we have these. Dominant for smoothness, but recessive for color, green. Then we have these. Dominant for color, but recessive being wrinkled. And finally, green wrinkled peas are the only 1 out of 16. So this is the ratio for dihybrid cross between the F1 plants that are heterozygous for each. So in other words, as we talked about before, alleles for each gene assort independently. So these are homologous pairs, and these are possible combinations of the distribution of those individual members of the homologous pairs after the meiosis. And this independent segregation, as we mentioned before, is due to the independent alignment of the homologous chromosomes on the equator of the dividing cell during the metaphase 1. So in other words, the genes for the color of the peas and the shape of the peas separate independently. In other words, the separation of the genes for the color does not affect the separation of the genes for the shape and vice versa. This independent assortment only works if the genes are located on different chromosomes. If genes are on the same chromosomes, they will be inherited together. This is called chromosomal linkage. So now, what if we don't know the genotype of an individual as dominant phenotype? In this case, we need to carry out the test cross. In this case, individual with unknown genotype is crossed with an individual who is or which is homozygous recessive. And then we can determine, based on the phenotypic ratios among offspring, the genotype of the unknown parent. For instance, here, if we cross, this is our test crossing plant, if we cross homozygous dominant with homozygous recessive, the outcome will be that all 4 plants will show dominant phenotype because all 4 of them will be heterozygous. But if we cross heterozygous dominant with homozygous recessive, half of the plants will have heterozygous dominant phenotype, and half of the plants will have homozygous recessive phenotype. So 50-50 ratio tells us that the unknown parent was heterozygous dominant. Now, Mendel's model of inheritance, it assumes that the trait is controlled by a single gene, and that each gene has only 2 alleles, and that there is a clear relationship in terms of dominant and recessive between these alleles. So Mendel's work was received with a bit of a skepticism. Some part of it is due to the fact that a lot of genes don't meet the criteria that Mendel applied to his experiments. For instance, in case of polygenic inheritance, multiple genes are involved in controlling the phenotype. So that phenotype is essentially an accumulation of the multiple genes contributions. It means that if there are multiple genes controlling the phenotype, you will see the continuous variation. These traits are referred to as quantitative. For example, human height is an example of a quantitative trait. So here, people with different height were placed on the football field, and you can see that kind of most people are in the middle. In other words, human height follows the bell curve, the normal distribution, with some median height being in the center. Now this is the skin color. Skin color is not black or white, it is a spectrum, so you can see that there are multiple genes that contribute to the distribution, the appearance of a person's skin color. Another example of non-Mendelian relationships is pleiotropy, when an allele have more than one effect on the phenotype. And pleiotropic effects are very difficult to predict. For instance, the gene SRY can be seen in diseases such as cystic fibrosis or sickle cell anemia. So multiple symptoms can be traced back to a single defective allele. For instance, in sickle cell anemia, the outcomes are the breakdown of red blood cells, or hemolysis, the clumping of cells and clogging of the vessels, accumulation of bad cells in the spleen, and that results in weakness, anemia, and heart failure, pain and fever, damage to the brain, splenomegaly and spleen damage, hepatomegaly, and damage to many other organs. And the outcomes are multiple, as you can see from this illustration. In some cases, more than 2 alleles can exist for a particular gene in the population. For instance, ABO blood type in humans is an example of multiple alleles. Each individual can have only 2 alleles, but the number of alleles is more than 2 here. So we're talking about outbreeding population. In other words, the population in which individuals breed freely and there is no inbreeding. So human blood type, ABO, is determined by the specific type of glycoproteins on the cell surface. If so, the IA is dominant than the I. So if somebody is homozygous for IA or heterozygous for IA, they're going to have blood type A. Somebody is homozygous for IB or heterozygous for IB, they're going to have blood type B. If they have both IA and IB, these two are not dominant over each other. It's called codominance. They're going to have blood type AB. And if they are homozygous recessive for II, they're going to have blood type O because there are no glycoproteins on the surface. And the type of the glycoprotein is determined by the single gene on the chromosome 9. And each individual, as we mentioned before, has two alleles of this gene. On the right, you can see the illustration of red blood cells. This is type A, this is type B, type AB, and type O. So how many alleles exist in the population? 3. How many alleles does an individual have? 2. So if an individual has 2 O alleles, type O, 2B or BO, type B, 2A or AO, type A, AB, type AB. So these are the combinations that we just discussed. And the main point is that there are 2 alleles, IA and IB, which are both dominant over the lowercase I, but they don't establish the dominance over each other. So in this case, A and B alleles are codominant. They display normal dominant recessive relationship with O allele, but neither of them is dominant over another. In other words, they are co-dominant. So, as we just saw, ABO blood type is determined by 3 alleles of a single gene. So we already explored codominance. There's also a phenomenon of incomplete dominance, in which the phenotype of heterozygote is intermediate between 2 homozygote phenotypes. So red and white flowers, hybrid will produce pink flowers. So here you can see red parent generation, white parent generation, cross-fertilization results in, and then cross-fertilization between the F1s results in 1 homozygous dominant, red, 1 homozygous recessive, white, and 2 heterozygous, which are pink. Environmental influence can also change the phenotypical presentation. So for instance, if the temperature of the fur of the Siamese cats is above 33 degrees Celsius, tyrosine is inactive and the fur has no color. But below 33 degrees Celsius, tyrosine is active, which allows them to produce melanin, which colors the hair black. It is also observed in Himalayan rabbits. Other environmental effects on the phenotype can include exercise and, to be honest, steroids, exposure to the sun, change your skin color, and things like diet and other life habits. Now, sex is also inherited. A lot of species, insects, birds, mammals, they have a set of sex chromosomes. And that set shown right here, will dictate the gender. Females have the pair of two X chromosomes. Males have the pair of XY. And sex chromosomes do segregate during meiosis, which means a gamete has either male or female chromosome. Chromosomes that are not sex chromosomes are called autosomes. So if you look at this, if this is the female parent, the gametes produced by the female will have one of two X chromosomes. This is the male parent, and the gametes will have either X or Y chromosome. When these gametes combine, there are two different outcomes. 50% of the offspring will be female, 50% of the offspring will be male. There are genes that exist exclusively on X or the Y chromosome. Genes that are carried on such sex chromosome are called sex-linked genes. X chromosome is bigger than Y and carries many more genes than Y. So you see the difference, 1,000 versus 78. And they have very few genes in common. They're not particularly similar. So females with the XX pair can be homozygous or heterozygous for a particular characteristic, while males will have only one copy of the genes found on the X chromosome. There will be no matching allele on the Y, which means there are diseases that linked straight to sex chromosomes. We call them sex-linked or X-linked diseases. For example, color blindness. Sex-linked diseases are more commonly seen in males because genes that are dysfunctional, genes for the disease, so to say, are found on X chromosome. Color blindness and hemophilia are striking examples. So this is the royalty, European royalty, and this is Queen Victoria, who was a carrier of the hemophilia gene and passed it down to the future generations, but only men in the future generation of European royalty was affected. So this is an example. It's a very interesting distribution. So remember, males cannot be carriers. They can only have hemophilia, but females can be carriers. So Queen Victoria was a carrier, and she married the Prince Albert. And two of their offspring established the present the British family. And you see that since Alexander of Denmark did not have the gene, and King Edward VII didn't have that gene either, the present British royalty is not affected. This couple, Duke of Albany, was actually hemophilic. Their daughter was a carrier, and their grandson was hemophilic. Louis IV, the Duke of Hesse-Darmstadt, and Alice, Princess of Hesse. Alice was the carrier, and that resulted in bringing that gene through the Tsarina Alexandra, who married the Tsar Nicholas II of Russia. Tsarevich Alexis, he was hemophilic. Beatrice and the Prince of Battenberg, Henry Prince of Battenberg, established essentially the Spanish royal house, which led to a bunch of offspring who were hemophiliacs. What you can see is that females, in this case, are not affected at all. They only can be carriers. Now, improper distribution of chromosomes during meiosis results in what's called aneuploidy. It initially happens during the anaphase 1 of meiosis 1 and results in either increased or decreased number of chromosomes in a particular gamete. The usual outcome is embryonic lethality, but in some cases, embryo can develop and survive beyond birth. And there are even people who live, yes, with this abnormal number of chromosomes. For instance, non-disjunction of sex chromosomes may result in a Turner syndrome when an offspring does not receive male or female, it's XO, only one female chromosome, trisomy X, 3 X chromosomes, Klinefelter syndrome, 2 X's and 1 Y. So you see abnormally high, abnormally low, and Jacob syndrome, XYY male. Now, non-disjunction abnormal autosomal number can occur in either father or mother during meiosis, and the probability of non-disjunction increases with age. If non-disjunction occurs, an egg has either 1 copy from a father or 3 copies, where 2 copies are either from mother or from father, of an autosomal chromosome. And the most striking example is trisomy 21, known as a Down syndrome. The 21st chromosome has 3 copies instead of 2. It happens 1 in about 900 births. That results in the symptoms such as weak muscle tone, partially open mouth, and distinctive eyelids. Also weakened immune system and heart problems and mental retardation. In our next chapter, we're going to talk about gene expression. So we're going to start with the discussion of the early ideas of how genes work. So most of the data came from the studying human diseases. Archibald Gorodin in 1902 realized that the disease called alkaptonuria inherited by the recessive allele, and he proposed that the patients who were sick did not have a particular enzyme. And it was the first idea that connected the genes to the enzymes. Now, Biddle and Tatum took it farther and decided to create mutations in chromosomes of a fungus, Neurospora crassa. They used X-rays to introduce DNA damage and look at the nutritional mutations. Essentially, they collected Neurospora crassa, mutagenized it with X-rays, grown the medium that contained all the nutrients, and then decided to grow it on so-called minimal medium. The mutants that did not grow on the minimal medium, but grew on the medium that contained amino acid arginine, were selected, because according to Biddle and Tatum, these mutants did not have the capacity to produce arginine. So essentially, they were trying to identify the fungal cells that could not produce specific enzymes. Since there are many biochemical pathways, Biddle and Tatum focused, as we mentioned before, on arginine. And they knew that the synthesis of arginine involves several enzymatic steps, and they managed to identify mutants that were deficient in each enzyme of that pathway. And they were the first to propose the one gene, one enzyme hypothesis, which was later modified to become one gene, one polypeptide hypothesis. So here you can see that the mutation in the enzyme E allowed the organisms that could not grow on the minimal medium grow in the minimal medium that had added ornithine, citrulline, arginosuccinate, or arginine. The mutation in the enzyme F did not allow fungi to grow on the medium with ornithine, but citrulline, arginosuccinate, or arginine was just fine. Mutation in the enzyme G, as you can see, prevented fungi from growing on the medium that contained either ornithine or citrulline, and mutation in the enzyme H allowed fungi to grow only on the medium that contained amino acid arginine. Based on this, they made the following conclusion: that the enzyme E, the very first one, facilitated the conversion of glutamate to ornithine. Enzyme F facilitated the conversion of ornithine to citrulline. Enzyme G facilitated the conversion of citrulline to arginosuccinate, and enzyme H facilitated the conversion of arginosuccinate to arginine. You can see from that pathway, if there is a mutation in the enzyme H, all previous enzymes are functional. But no matter whether you have these chemicals or not, the only chemical that cannot be produced is arginine, because enzyme H is affected. So cells can only grow if arginine is present. And that led them to the conclusion that essentially a single gene encodes for a single enzyme, as we mentioned before. Now, in the next several minutes of conversation, we're going to try to establish the link between the genes and the proteins, pretty much. So proteins, as we discussed in the class in our cookbook analogy, are the molecules that essentially control most of the cell functions, such as shape, reproduction, synthesis, enzymes, signaling, you name it. Now, the information that is contained within the genes of the DNA has to be linked to the proteins that essentially run the cells. So new molecule synthesis within the cell occurs through the biochemical pathways, and each step in that biochemical pathway is facilitated by an enzyme. And as we mentioned before, Biddle and Tatum managed to demonstrate that the knockout or mutation in a particular gene results in the dysfunctional enzyme that's encoded by that gene. Now, central dogma of molecular biology was first described by Francis Crick in the interview to a journalist, and according to Crick, he was very, very upset that it turned out to be a dogma. Crick basically explained that the information in the cell flows from the DNA to RNA to protein. So the process of synthesis of RNA using DNA as a template is called transcription, while the process of the synthesis of protein using RNA as the template is called translation. Now, why is this bad idea to call it a dogma? Retroviruses can use single-stranded RNA as a template to produce double-stranded DNA in the process called reverse transcription. This process eventually found a lot of practical applications. So, as we mentioned before, transcription is the synthesis of RNA that is directed by DNA template. Only one template strand of DNA is used, but both strands have to be present. Now, another difference is that in DNA there is a thymine. In RNA, that will be uracil. So this is the replacement. This is the difference between the DNA and RNA in terms of the composition of nucleotides. Now, product of transcription is mRNA, and the function of mRNA is to direct synthesis of polypeptides/proteins. During translation, proteins are synthesized, polypeptides that are later shaped into proteins. Translation takes place on the ribosome and requires multiple types of RNA. Now, why is that so? So DNA in eukaryotes is confined to the nucleus, while the protein synthesis in eukaryotes occurs at the ribosomes. Ribosomes are located, for the most part, in rough endoplasmic reticulum or in the cytoplasm, which means that DNA information has to be carried from the nucleus to ribosomes by the intermediary RNA molecule called messenger RNA. So RNA is synthesized from DNA in the process of transcription. And the transcription results in the formation of messenger RNA, mRNA, that carries the message from the nucleus to the ribosomes, ribosomal RNA, which is one of the key structural and functional components of ribosomes, and transfer RNA, which carries amino acids to the site of translation. Now, before we proceed, I need to highlight the main differences between the DNA and RNA. Instead of T, RNA has uracil. Instead of deoxyribose, RNA has ribose. And DNA is double-stranded, while RNA, for our intents and purposes, has only one strand. So this is the example of the functions of those three molecules. So messenger RNA, or mRNA, carries the information of the amino acid sequence of a protein to the ribosome. Ribosome contains ribosomal RNA, which carry out the process of peptide bond formation between amino acids during protein synthesis. And finally, transfer RNA carries amino acids to the site of the protein synthesis on the ribosome, which ensures that the correct amino acid is incorporated into the protein. Now, I had a bit of a glitch with the computer recording, so I'm just going to talk about tRNA again. It carries the, and I apologize if there's a gap in the recording. So again, transfer RNA carries the attached amino acid to the site of the protein synthesis on the ribosome to ensure that the correct amino acid is incorporated into the proteins. So now let's talk about the genetic code. So genetic code is organized in codons. Each codon is the block of 3 DNA nucleotides that correspond to a single amino acid. It's important to understand that a codon can encode only one amino acid. Now, one of the key concepts in genetic code is the concept of the reading frame. When a ribosome reads the code, it reads 3 letters. It doesn't, so the codons never overlap. It's like individual words in the language. It was identified when scientists introduced insertions and deletions and looked for the changes in amino acid sequence of the polypeptide. Those insertions or deletions in the sequence of the DNA or RNA are called frameshift mutations. So here you can see the example of the genetic code. Each of these codons encodes for a single amino acid. Now here you can see that if you change the codon ACG into the codon CAC, you change an amino acid from threonine to histidine. Okay? You delete this ACG, essentially. Now in this case, so this is the frameshift mutation. It's a deletion mutation, I'm sorry. On the bottom, you can see the deletion of one nucleotide results in the dramatic shift of the reading frame. Okay, so now if you look at the previous picture, the only thing that was achieved by removing 3 nucleotides is that amino acid threonine is gone, but the rest of the sequence of amino acids is the same. But if you remove just one nucleotide, then the sequence of amino acids dramatically changes. So as we mentioned before, in the genetic code, the codons specify amino acids, which means one codon specifies only one amino acid, and there are so-called stop codons, which stop the translation. There is a single start codon, which starts the translation. And a very important feature of the genetic code is that it is a degenerate code, which means several amino acids can be specified by more than one codon. So this is the table of the genetic code. I'm not going to go over it, and I'm not asking you to memorize any of this. I just want you to understand this. For instance, proline can be encoded by 4 codons. Leucine can be encoded by 6 different codons. There are amino acids that are encoded by only one codon, methionine, for example, and tryptophan, but many amino acids, as you can see, are encoded by more than one. So the genetic code is practically universal. Oh yeah, and the feature that the genetic code, in the genetic code, a single amino acid can be encoded by more than one codon means that the code is degenerate. Now, code is universal. Why? Because all living things share the common ancestry. Okay, so that's why it is universal. And interestingly enough, mitochondrial chloroplasts, while they have, remember, they do have DNA, but while they have ability to produce proteins, they have ribosomes. The genetic code in mitochondria and chloroplast is a little different. The stop codons are slightly different. The reason for that, you may remember, mitochondria and chloroplasts, they evolved, they become endosymbiotic. They used to be bacteria. So the code in bacteria and the code in eukaryotes is tiny, slightly different. So let's get back to the gene expression. Essentially, there are two steps in gene expression: transcription and translation. During transcription, the information in a gene is copied into messenger RNA. During translation, protein is synthesized using mRNA as the template. tRNA, amino acids, and ribosomes are required for the translation to occur. It's important to remember that essentially the sequence of the protein is determined by the sequence of the nucleotides in the gene. So this is just another summary. Let's go over it. So during transcription, a short segment of one DNA strand is transcribed into one RNA molecule, either mRNA, tRNA, or rRNA. The enzyme that's responsible is RNA polymerase. And the base pairing is between DNA and RNA. During translation, mRNA is used as the template to produce a single protein molecule. Ribosome is required to facilitate that process. And the pairing that is essential is between messenger and tRNA. The codon in mRNA must form the base pairs with anti-codon in tRNA. Now, I want you to notice the difference between the replication and transcription. In replication, the template is the DNA, but the molecule that is produced is also DNA. Two double helices are produced during the replication. The enzyme during replication, unlike during transcription, is DNA polymerase, and the pairing is between two DNA molecules. So now we're going to talk about transcription. Transcription consists of three main steps: initiation, elongation, and termination. During initiation, the strands of DNA molecule unwind and separated at the beginning of the sequence of the gene. The enzyme that facilitates the process, RNA polymerase, binds to the sequence known as promoter. And this promoter region located specifically at what's called the template strand. DNA, as you remember, consists of two strands. Only one of them serves as the template. Now, during elongation, RNA polymerase produces a sequence of RNA nucleotides using DNA as a template, and importantly, it uses only one strand of the DNA. So during that synthesis, bases in the RNA strand that is synthesized from scratch are complementary to the bases in the template strand of DNA. Eventually, RNA strand peels away from DNA template, and DNA strands that were separated before now repair and wind up forming the double helix again. So here you can see that RNA polymerase during elongation produces an RNA molecule. And since this RNA polymerase moves to the right, you can see that these DNA strands are annealing back and wound up being a double helix again. Now, this is the image of the RNA synthesis on the DNA under the microscope. And you can see that there are multiple RNA polymerases moving along the DNA molecule, producing all those long chains of RNA. So it tells you that the synthesis of RNA, the transcription, is highly efficient. It happens at multiple sites at once, and many RNA molecules are produced. Transcription termination occurs when RNA polymerase reaches the termination signal. At this point, it releases the newly synthesized RNA molecule, and DNA strand is completely restored. It again forms the double helix. RNA polymerase is released from the DNA and can start another round of transcription again. So at the end of transcription, DNA double helix remains intact. RNA is in some cases ready to be translated, and RNA polymerase is ready to start the new round of transcription again. Now, messenger RNA, which is the product of transcription, is the intermediate necessary to convey the gene sequence of the genomic DNA to the ribosome. It essentially serves as a complementary copy of a gene, and ribosome reads that complementary copy of the gene. Now, it's important to remember about the differences between pro and eukaryotes. In prokaryotes, chromosomes are not found in nucleus, but are rather found in the cytoplasm. And the genes that are responsible for the same function usually clustered together. All nucleotides in the gene encode for amino acids. There are no junk DNA. And that explains why transcription and translation in prokaryotes occur practically simultaneously in the same cellular compartment, in other words, in the cytoplasm. Here, you can see a molecule of DNA. These are RNA molecules, and these are ribosomes. So you can see that RNA molecules are still being synthesized. But while they are being synthesized, ribosomes already bind to them and produce proteins. So this illustrates it even further. Here you can see the DNA molecule in the center. DNA is being transcribed by RNA polymerase, producing RNA, and RNA immediately is translated by ribosomes, producing protein. Now, what about the transcription in eukaryotes? Well, it is a little bit more complicated. First of all, in eukaryotes, transcription is carried out by 3 different RNA polymerases. RNA polymerase 1 synthesizes ribosomal RNA. RNA polymerase 2 synthesizes messenger RNA. And RNA polymerase 3 synthesizes transfer RNA and some other small RNA molecules. Each type of RNA recognizes its own promoter. Unlike in prokaryotes, in eukaryotes, the initiation of transcription requires a series of transcription factors. They are essential to get RNA polymerase 2 to bind to the promoter and to initiate the gene expression. Transcription factors interact with RNA polymerase, forming initiation complex that binds to the promoter. And the termination in eukaryotic cells is also much less defined. So here you can see transcription factors binding to RNA polymerase, which is transcription factors binding to some other transcription factors, which already are at the promoter. When they form that complex, they recruit RNA polymerase II, which forms the initiation complex, which is ready for transcription. Another difference between eu and prokaryotes is that in eukaryotes, mRNA has to be modified. Primary transcript has to have 5' cap added. Cap structure is added to the 5' end of RNA molecule. This cap structure protects mRNA from degradation, and it also is essential for the initiation of translation. Poly A tail consisting of multiple adenine nucleotides, is added to the 3' end of RNA synthesized during transcription, and the main purpose of it is to protect RNA from degradation. Also, it facilitates the transport of mRNA from the nucleus. And finally, the non-coding sequences, introns, have to be removed from the RNA. So this is the big difference between eukaryotic and prokaryotic transcription. In prokaryotes, as we mentioned before, in the DNA, all nucleotides encode for proteins. In eukaryotes, it's not true. There are large chunks of DNA which do not encode for any proteins. So here you can see the modifications. There is a 5' cap, so this is the cap structure, please don't memorize it. This is the 3' poly A tail. Now, the transcription and translation in eukaryotes are separated in time, as we will see, and in space. DNA is in the nucleus, so transcription in eukaryotes occurs in the nucleus. Ribosomes, on the other hand, are in the cytoplasm. So the protein synthesis occurs in a cytoplasm or on the endoplasmic reticulum. In addition to that, genes that encode proteins for the same biochemical pathway aren't clustered together in the same chromosome and can be dispersed throughout the genome. Finally, the segments of the DNA that encode for the protein, so-called exons, are separated in eukaryotes by segments that are not encoding, introns. So when DNA is transcribed in eukaryotes, the transcript contains both introns and exons. So enzymes that are present in the nucleus form the structure called spliceosome, and these enzymes essentially cut out introns and stitch or splice together exons to form the final product called mRNA. And then mRNA exits the nucleus through the pores in the nuclear membrane and forms the complex with a ribosome. So the process of excision of introns out of the primary transcript is called splicing. Here you can see the structure of the eukaryotic gene. There is a promoter, there are exons separated by non-coding introns. So-called ribonucleoprotein particles, or RNPs, recognize the boundaries between the introns and exons, cluster with each other, and form spliceosome, which removes introns. So essentially, look at this. When DNA template is transcribed, it forms primary transcript, which already has 5' cap after modification and poly A tail, but it also contains introns. Then, during the process of splicing, when introns are removed, eukaryotes finally have the mature mRNA with a 5' cap and 3' poly A tail. Splicing provides an opportunity for an alternative splicing, when a primary transcript can be spliced into different mRNA by including different sets of exons. So for instance, if there are 5 exons, 1, 2, 3, 4, 5, the final product, final mRNA can, for instance, include 1, 2, 3, and 5, but not 4. So that allows cells to make multiple proteins with slightly different functions from the single gene. Not only it benefits humans, it also may affect humans because it may lead to a variety of genetic disorders because of that altered splicing. A lot of human genes, they actually exhibit some form of the alternative splicing, and it essentially explains how 25,000 genes in the human genome can encode more than 80,000 different mRNA. Imagine you have a gene, and as the result of alternative splicing, a single gene produces 3 or 4 different versions of the mRNA. 1, 2, 3, 5, 1, 2, 4, 1, 2, 4, 5, 2, 3, 5, and so on and so forth. There are many variants that can exist. Now we're going to talk about the translation of mRNA into a protein. The function of transfer RNA molecules is to carry amino acids to the ribosomes. So these amino acids can be incorporated into the peptides. Amino acids are added to the acceptor stem of transfer RNA molecule. On the other end of the molecule is anticodon loop, which contains 3 nucleotides complementary to one of 60 mRNA codons. Ribosomes, another critical component for protein synthesis, are composed of 2 subunits, small and large. Small subunit binds mRNA and tRNA. Large subunit has binding sites for 2 other tRNA molecules and the catalytic site, which is responsible for forming peptide bond between amino acids during the protein synthesis. tRNA essentially binds chemically amino acids and brings amino acid to a ribosome. At least one type of tRNA is assigned to carry each of the 20 different amino acids. So this is the illustration of the transfer RNA. Here is the anticodon loop, essentially 3 bases that are complementary to a codon. And each transfer RNA has the acceptor end that carries an amino acid. Now, it's important to understand that the bases of the anticodon loop pair with mRNA codon inside of the ribosome binding site. And tRNA is charged. During charging reaction, amino acid is added to the tRNA molecule using energy in the form of ATP. It means that when tRNA binds to the ribosome and the peptide bond is formed, peptide bond can be formed without any additional energy because energy has been spent on adding an amino acid, say tryptophan, to the tRNA. It's important to understand that ribosomes do not verify an amino acid that is attached to transfer RNA. Instead, the match between the anticodon and codon ensures that the proper amino acid is introduced into the protein sequence. So, if you look at the entire picture, this is the tRNA and this is an amino acid that's added to the amino acid site. This tRNA is now charged. This tRNA binds to the accepting site on the ribosome. Adenosine monophosphate is released from the complex, and now charged tRNA is ready to travel to the ribosome. So this illustration shows you the association of the ribosome with the RNA. You can clearly see that there are three sites on the ribosome: A, accepting or attachment; B, polypeptide or protein; and E, exit sites. So ribosome has two primary functions. One of them is to decode mRNA. Another is to form peptide bonds. So during translation, ribosomes transfer and messenger RNA cooperate to synthesize proteins, and translation begins with the process of initiation. mRNA binds to the small ribosomal subunit, and then mRNA slides through that subunit until the first start codon, AUG, is exposed in the first tRNA binding site. First tRNA that carries an amino acid methionine and has an anticodon AUC, which is complementary to the codon AUG, that's the codon for methionine. It binds to mRNA start codon and it completes initiation complex. Now, large ribosomal subunit joins the party, and it's all set. Small ribosomal subunit, methionine tRNA with methionine amino acid bound to UAC codon, and large subunit joins the party. So you can see that tRNA is now in the first tRNA binding site. What happens next? So in elongation, second tRNA binds to second binding site, which is adjacent to the first. How? Well, anticodon of second tRNA is complementary to the codon on mRNA that is now exposed in a second tRNA binding site. When both of these tRNAs are bound, peptide bond forms between methionine and a second amino acid. This is facilitated by the ribosome catalytic site, and first amino acid is released from first amino acid, which is methionine in this case, is released from the tRNA in the first binding site, and empty tRNA, remember, tRNA that doesn't have an amino acid bound anymore, leaves ribosome. Ribosome translocates, moves down mRNA by one codon, and it transfers tRNA that holds now 2 amino acid chain to the first binding site. New tRNA, basically number 3, with anticodon that is complementary to newly exposed codon in the second tRNA binding site, approaches and the cycle repeats. tRNAs that are empty and don't have amino acids bound to them anymore get reloaded with appropriate amino acids in the cytoplasm. So this is methionine in the first binding site. This is valin tRNA in the second. Methionine from the first binding site is transferred to valin, forming the peptide bond. Ribosome shifts, so now tRNA with a growing polypeptide chain is in the first binding site. Second is empty. Now, this process goes on until ribosome runs into the stop codon, which slides into second tRNA binding site. Special protein called release factor binds to the stop codon. Ribosome separates into subunits. Protein chain is released. mRNA is released, and importantly, it's not degraded. It can be used to make another protein. So look at this. Histidine receives the dipeptide from the first binding site. It's all going and going and going until the ribosome hits the stop codon. At stop codon, ribosome disassembles and polypeptide chain is finally released. Now let's review the whole process. So in eukaryotes, RNA polymerase II in the nucleus copies only one strand of DNA, the template strand, producing the primary transcript. The primary transcript is processed. 5' cap is added, poly A tail is added to the 3' end, and introns are removed, which produces mature mRNA. This mature mRNA is exported from the nucleus through the pores into the cytoplasm. 5' cap of mRNA associates with small subunit of the ribosome, and large subunit is finally added to the initiation complex. Now ribosome cycle begins when the growing peptide is attached to the tRNA in the P site. The new tRNA approaches the A site, the polypeptide is transferred to the incoming tRNA, and the chain keeps growing. Ribosome shifts every time the new ribosome cycle starts, and the empty tRNAs are released from the ribosome to get amino acids attached to them again and again and again and bring them to the ribosome. When translocation of the ribosome meets stop codon, polypeptide chain is released. So now let's mention a few differences between pro and eukaryotic gene expression. In prokaryotes, there are no introns. In prokaryotes, several genes can be transcribed into a single mRNA molecule, while in eukaryotes only one gene per mRNA can exist. Transcription and translation in prokaryotes are coupled, they happen at the same time, while in eukaryotes, transcription and translation is separated in space. Transcription is in the nucleus, translation is in the cytoplasm, and in time. Transcription has to be completed first before translation starts. Initiation in both of them starts at AUG codon, but in eukaryotes, ribosomes have to recognize 5' cap. mRNA after transcription in prokaryotes does not have to be modified, but it has to be extensively modified in eukaryotes by the addition of 5' cap, 3' poly A tail, and the removal of introns. So this is just a recap of what we talked about, that in eukaryotes, the gene codes for a single protein, in prokaryotes as well. Transcription produces mRNA that is complementary to the DNA gene, template strand. mRNA strand associates with the ribosome, and tRNA in the cytoplasm bring amino acids to the ribosome. Ribosome selects tRNA based on the pairing between anticodon and codon, and the start and stop signals define where protein synthesis starts and where it ends. This is just another illustration of the same concept. I'm not going to spend time on this, but it's a good illustration of what's going on during transcription and translation in eukaryotes.